Hello and welcome to Easy Learning with Dr. Salman Khan. In this video, we will be studying the parasite TRYPANOSOMA brucei. These are the sections under which we will be studying: Introduction, Epidemiology, Habitat, Morphology, Life Cycle, Modes of Infection, Pathogenesis, Clinical Features, Laboratory Diagnosis, and Prophylaxis. First, introduction to the parasite. The word trypanosoma comes from the word trypanes, which means to bore, and soma, which refers to body. All members of the genus Trypanosoma exist at some time in their life cycle as trypomastigote stage with an elongated spindle-shaped body, central nucleus, a posterior kinetoplast, and a long undulating membrane. Volutin granules are found in cytoplasm. Some trypanosomes, such as Trypanosoma brucei, assume amastigote forms in vertebrate hosts. In addition to the typical forms, cells with atypical features are frequently found, a condition known as polymorphism. Trypanosomes infecting man: first, Trypanosoma brucei complex, which causes African trypanosomiasis or sleeping sickness. The subspecies are Trypanosoma brucei gambians, which causes West African sleeping sickness. Then Trypanosoma brucei rhodesians, it causes East African sleeping sickness. Second, Trypanosoma cruzi, which causes South American trypanosomiasis or Chagas disease. And the third is Trypanosoma rangeli, a non-pathogenic trypanosome causing human infection in South America. Then introduction to the parasite. It can be defined as the parasite causing African trypanosomiasis in man. It is one of the earliest protozoan parasites to have been recorded. Then the history. Trypanosome was first isolated from the blood of a steamboat captain on the Gambia River in the year 1901, hence the name Gambians by Ford. Then Dutton in the year 1902 proposed the name Trypanosoma gambians. For the next subspecies, it was discovered by Stephens and Fantham in Rhodesia in the year 1909, hence the name Trypanosoma brucei rhodesians. Next, epidemiology. Trypanosomiasis is believed to have been existing in tropical Africa from antiquity. It is endemic in scattered foci in West and Central Africa between 15 degrees and 18 degrees south latitudes. Then the habitat. Trypanosomes live in man and other vertebrate hosts. They are essentially a parasite of connective tissue where they multiply rapidly and then invade regional lymph nodes, blood, and finally may involve a central nervous system. Then morphology. It is divided into vertebrate form and insect forms. Vertebrate form. In the blood of vertebrate host, Trypanosoma brucei gambians exists as trypomastigote form. Then insect forms. In insects, it occurs in two forms. First is epimastigotes, second is metacyclic trypomastigote forms. First, about the trypomastigote form. In the blood of vertebrate host, Trypanosoma brucei gambians exists as trypomastigote form, which is highly pleomorphic. It occurs as a long slender form, a stumpy short broad form with attenuated or absent flagellum, and an intermediate form. The trypomastigotes are about 15 to 40 micrometers long and 1.5 to 3.5 micrometers broad. In fresh blood films, trypomastigotes are seen as colorless, spindle-shaped bodies that move rapidly, spinning around the red cells. In smears stained with Giemsa or other Romanovsky's stain, the cytoplasm appears pale blue and the nucleus appears red. The kinetoplast appears as a deep red dot and volutin granules stain deep blue. The undulating membrane appears pale blue and the flagellum red. This is the diagram of the trypomastigote with the structures it contains. It is spindle-shaped organism with a blunted posterior end and a finely pointed anterior end. The nucleus is large, oval, and central in position. The kinetoplast is small and is situated at the posterior end. The flagellum starts from the posterior extremity near the kinetoplast, curves around the body in the form of an undulating membrane, and then continues beyond the anterior end as a free flagellum. The undulating membrane is thrown into folds, the number of which, usually 3 to 4, varies with the length of the parasite. It is an actively motile flagellate. Next, polymorphism. The trypomastigotes of Trypanosoma brucei are polymorphic. Individual parasites vary in size and shape in the different stages of their existence. Two main forms are recognized. The one, short, thick, stumpy form, which is 10 micrometers long by 5 micrometers broad with no free flagellum or having only a very small one. The other, long, slender form, which is 20 micrometers long by 3 micrometers broad, having a conspicuously long free flagellum. Between these two forms, intermediate forms are occasionally observed. The first form to appear is a long, slender one, and later, when the infection is established, the short, stumpy form together with the intermediate one appear. Then antigenic variation. The parasite has an antigenic variant. A succession of variants occur in Trypanosoma brucei infection both in animals, that is rabbits, and man. Each new variant emerges at 3 to 4 days intervals, possibly by selection of mutants. This appears to depend upon the ability of the host's defense mechanism, and when the defenses fail, the final variant multiplies unchecked. Each wave of parasitemia is accompanied by fever and followed by leukocytosis, specifically monocytosis, but not neutrophilia. It may be that the outer proteinaceous coat may protect the organism in escaping from the host's defense mechanism. Another feature is that the antibodies develop to a series of immunologically distinct type variant in the serum of hosts. Each rise in antibody titer coincides with the disappearance of the homologous variant, and when this has been eliminated, a new antigenic type immediately arises. Trypanosomes exhibit unique antigenic variation of their glycoproteins. Each successive wave represents a variant antigenic type VAT of trypomastidode possessing variant-specific surface antigens, that is VSSAs, or variant surface glycoprotein VSG coat antigen. It is estimated that a single trypanosome may have as many as 1000 or more VSG genes that help to evade immune response. Besides this, trypanosomes also have other mechanisms that help them to evade host immune responses. This is the parasite when seen under a microscope, and this is when it is stained. Then life cycle. CC fly is the intermediate host, whereas human is the definitive host. First, development in CC fly. In the midgut of the fly, short stumpy trypomastigotes develop into long slender forms and multiply. After 2 to 3 weeks, they migrate to the salivary glands, where they develop into epimastigotes, which multiply and fill the cavity of the gland and eventually transform into the infective metacyclic trypomastigotes. Development of the infective stage within the CC fly requires 25 to 50 days. This is called as extrinsic incubation period. Thereafter, the fly remains infective throughout its life of about 6 months. Then, development in man and other vertebrate hosts. Metacyclic stage, infective form of trypomastigotes are inoculated into a man, the definitive host, through the skin when an infected CC fly takes a blood meal. The parasite transforms into slender forms that multiply asexually for 1 to 2 days before entering the peripheral blood and lymphatic circulation. These become stumpy via intermediate forms and enter the bloodstream. In chronic infection, the parasite invades the central nervous system. Trypomastigotes, which are short, plumpy form, are ingested by the CC fly, male or female, during a blood meal. Then, next, modes of infection. The source infective form is the metacyclic trypomastigote form, which are infective to humans. Route through the skin by the bite of CC fly, and congenital transmission has also been recorded. Vehicle is the CC fly. If you wish to see more of such videos, do comment below. And don't forget to like, share and subscribe to my channel. Also, press the bell icon for early notification of my new videos.